Ladies and gentlemen, welcome to special lecture. Today our invited speaker is Dr. Meunier. Before his speech, Vice President Yu Wang will give us the welcome remark and introduction of Dr. Meunier. Then to welcome Vice President Wang.
Well, dear colleagues, it's my great honor to introduce our distinguished speaker today. President Meunier is right now the president of Academy of Science in France. I guess his title are stated on the poster here. He was trained as a coordination chemist. So he was located at Lab of Coordination Chemistry in Toulouse, France. And then that's his past interest, and then he switched to the drug development, which is closely related with chemistry, I believe. He started his presidency from last year, 15, and before that he was vice president of Academy of Science in France. He was the president of CNRS, which is a famous research organization, I would say, because most of CNRS is not like our academy. There's only a few laboratory. One of them are Laboratory of Coordination Chemistry, which is just like one of the institutes in academy. The other CNRS are distributed among the universities, which we thought it was a very nice system to mingle with university faculties and jointly train our young scientists. Well, as I said, his present interest was design and develop drug to be the treatment of a certain disease, which is there was an organization called PALUMED, and he's the founder of this organization and member of the board since December 2000, and they're dealing with malaria, bacteria infection, and also Alzheimer disease, which is very important for the society. And I guess I couldn't say much about all the list, but just a few. He was the member of French Academy of Science, of course, since 1999, president of CNRS 2004 to 2006, foreign member of Polish Academy of Science 2005, and I just mentioned 2013 to 2014. He was vice president of French Academy of Science, and now he's the president of the Academy of Science. And he obtained many awards, and just name a few in recent years. Humboldt-Galusek, this is a famous chemist. He won the prize in 2002. And Knight in Order of Legion of Honor, which you see the pictures on the poster in there, and that's the ceremony of this award. And also prize Label of French Chemical Society in 2007. So with this, let's welcome President Meunier. Looking forward.
It's okay, say. Thank you. Dear Professor Wong, I would like to thank you for your very kind invitation. It's a pleasure to be here in Taipei. It's my second visit. I was in Taipei about more than 15 years ago and giving a talk in one of the Department of Chemistry of the Academia Sinica. So it's always a pleasure. So I'm back now as a president. It's also a pleasure to have this direct contact at this level between our two academies. For the French Academy, the Academia Sinica is one of the favorite academy, I would like to say. Concerning the involvement of chemistry with drug development, I would like to say one thing. We have a tendency to forget that 80% of the new drugs are still based on chemicals. 20% are based on the so-called biopharmaceutical, that's mainly antibodies or recombinant proteins. But still, if you look to the data, it's based on the statistic of the FDA, American Agency for Food and Drug Administration. We have to consider that chemistry has been involved and used to be involved and will be involved in the future for drug development. So we don't have to forget chemistry when we are talking about drug. Today I would like to speak about Alzheimer's disease and the challenge of trying to make new drugs for Alzheimer's disease. It's a very challenging area. Let's start with one slide concerning this disease. It's a disease related to aging. If you consider the evolution of life expectancy in France, for example, since the 50s, this in red, the evolution of the life expectancy for men. That's for female. Okay, here, sorry, in green. And this is a black zone for Alzheimer's disease. Most of the people who get sporadic disease, that's mean the disease will not related to a genetic problem. Only 2% of Alzheimer's disease is related to genetics. Some people, it's the so-called the family Alzheimer's disease, which correspond to some gene modification for 2% of the people who get this Alzheimer's disease, and usually they get the disease at the age of 50 or 60. But for other people, usually they get the disease where they're around between 75 and 80. So this is a bad zone. If you don't get disease at this age and you pass this zone, then you feel happy and you will survive for the rest of your life without problem with Alzheimer's disease. And you see that there is a saying. People say female has more tendency to get Alzheimer's disease than male. This is just because the life expectancy is different for male and female, and you see that the female gets the disease before man. One point. The retirement age in France in the 50s was 65, and most of people died before. So there was no problem with retirement pension. Now it's different. You can see that's a gap, but that's a statistic and demographic problem. This is a real fact. Now, Alzheimer's disease, as I say, is a disease related to aging, not related to genetics. We can probably talk about gene of predisposition for the disease, but the key factor is the aging. And there are some figures which I speak about. Alzheimer is not a digenetic or trigenetic disease. It's results of modification of the expression of many different proteins, especially post-transcription modification. So we can talk about the role of microRNA, post-traduction modification, glycation of proteins. We know that we have, when we get older and older, we have a glycation of hemoglobin, for example. And that's a way, if we have too much of glycation of hemoglobin, that's not very good. We have diabetes and something like that. So we have to consider this is not a mono, di, or trigenetic disease. And I will come back on that point when I will talk about animal models for Alzheimer's disease. Neuron, synapse, and brain. We have to remember that the fetus is producing 8 neurons per second in utero. Huge production of neurons. A normal brain contains about 100 billions of neurons. 100 billions of neurons. But the key point is also the connection between neurons. That's the synapse. We have 10 to the 14, 10 to the 15 synapse. In each cubic millimeter of brain, we have 1 billion of synapses. And the key point is to remember that brain is a powerful molecular computer. When we think about molecular computer, we have to think back to brain. Our brain is a wonderful molecular computer. And nobody can compete with a brain. People are dreaming about having computers which are able to compete with the brain. And I will tell you a saying by colleagues from the Academy of Science about a computer. We will have computer able to compete to brain when we will have computer which will be able to laugh about a joke that it didn't know the day before. So that's the difference between the brain and a computer. Computer is good to do things that the brain is slow to do things, but we have difference between a computer and a brain. Okay, now aging and brain. The normal aging process for brain, for everybody, is about the fact that we are losing 10% of our neurons where we are getting older and older. 10%. That's a normal aging of brain. So we start a maximum of neurons on brain on synapse connection when we run around 20. And then we start to have a decrease of the number of neurons. That's the aging. Well, but when at my age, I have less neurons than I was young. But there is a key difference between my situation at my age and when I was 20. Because I know now if I have a problem with 3 different solutions, because my experience, I will be able to choose just the right solution. When you are 20, you want to try everything. That's the difference. So even if I have 10% neuron less, I'm still able to do some research. And we have to think about brain exercise. Brain exercise is a good point, because if you have a good brain training, that means you are going to have a good neuronal plasticity, which increase the brain activity when you are doing a good activity with your brain. And if you stop using your brain, you decrease the connection between the neurons. So you build the network with activity, and you destroy the network because you reduce the activity. So we have to use our brain and to be as best as possible, as good as possible for the use of the brain. And there is a tendency in the new education system to say, don't use the brain because you are going to, don't learn things by art, don't learn, you have just to understand the situation, things like that. It's just a stupid system. Because we need to have a brain functioning like a good engine system in order to build the neuronal plasticity. And I'm saying that not because I'm a specialist about neuron. I'm just saying that because Jean-Pierre Changeux is a French specialist about neurons. What do we have to cure Alzheimer's disease at the moment? A short list of bad drugs. These are the drugs. Most are inhibitors of cholineacetylesterase. There is one antagonist of the non-competitive agonist of the N-methyl D-aspartate receptor, memantine. So these are drugs on the market for the last 20 or 30 years, and they are not very good. They are unable to stop the evolution of the disease. They are reducing some symptoms, but strictly speaking, these drugs should not be on the market because they are unable to do something with the disease. They are unable to reduce the disease, to stop the evolution of the disease, but we have nothing else at the moment. The situation is very bad, and the challenge for making new drugs is real challenge compared to other sector of the disease. Number of people is increasing. For example, we can imagine that the number of people having the disease worldwide will be 100 million in the near future. And the key point is from 2002 to 2012, 99.4% of the clinical disease for Alzheimer's disease drug failed. And if you remove from this period memantine, which has been filed as a drug by FDA in 2003, then you go to 100%. 100% of failure within a period of 10 years. That's a real disaster. 413 Alzheimer's disease clinical trials have been performed during this period, including 83 phase 3 trials at the last stage. All these clinical trials failed. That means big companies are not very happy when you are talking about Alzheimer's disease project. They say, no chance to get a drug. We spend billions of dollars on that topics in that domain for the last 15 years with nothing. So you see that there is a real reduction of clinical trials concerning Alzheimer's disease. For example, there is only at the moment nearly 100 clinical trials compared to the situation for anti-cancer drugs. 14 times more clinical trials on the field of anti-cancer drugs compared to Alzheimer's disease. Why is that? Because at the moment, if you are running clinical trials on cancer, you have 20% chance to get a new drug when you enter in clinical trials. 20% in a field where you have a huge amount of real good drugs. That means the success, the chance to get a drug at the end is not too bad. For Alzheimer's disease, it's nothing. So very challenging at the moment. There are three categories of drug candidate for Alzheimer's disease. The molecules which may be able to improve, this one is definitively dead. No? Okay. Able to improve the symptoms, okay, able to improve cognition. And there are maybe some candidates, I'm not too good, as disease-modifying drugs. That means that will be the magic drug, the drugs which will be able to stop the evolution of the disease in the early stage of the disease. We don't need drugs which are able to stop the evolution of the disease at the end of the disease. AD period, the period for Alzheimer's disease is from 6 to 10 years. But nobody wants to have a drug to get 4 or 5 more bad years of the disease. We need a drug which will be able to stop the evolution of the disease just at the beginning. When we know, when we will be able to have the biomarker to understand, oh, That's the beginning. In order to stop the evolution of the disease and to die from heart attack, whatever it is, 20 years after. People are talking also about antibodies, immunotherapy for Alzheimer's disease. Frankly speaking, antibodies are not really able to cross the BBB, the blood-brain barrier. How to do something where you are outside of the BBB when the problem of the disease is on the other side of the BBB. I'm not sure that's a real future for antibodies. Antibodies are wonderful biological tools or biopharmaceutical for cancer, for example, because antibodies are able to distinguish between normal cells and cancer cells. But concerning Alzheimer's disease, which is a brain disease, I'm not sure that's a real future. I may be wrong, but that's my current opinion at the moment. What shall we do concerning Alzheimer's disease? We know that many pharmaceutical companies decided to stop research, and they are waiting for new drugs after phase 2. Usually when you talk about Alzheimer's disease with pharmaceutical company, they say, well, when you will be at the end of phase 2, then we will buy your molecule. But who will be able to go to phase 2 without the support of a strong and powerful pharmaceutical company? That's a challenge in the field. So many people, many researchers decided just to describe the disease. That's an easy approach. You just talk about the disease. No way to fail, because you just describe the disease. But you can make good publication from that. And many people are working on biomarker to predict the disease. It's a wonderful area. It's relatively easy to have a biomarker on the market because the barrier to get a drug approval is not the same high than for a real drug. But if we get biomarker without drug, that's very depressing. I will be very uncomfortable to go to a medical doctor and say, well, now I can tell you, in 7 years, you have 100% chance to get Alzheimer's disease. At the moment, I don't know. Maybe, maybe not. But what I know, I'm at the moment enjoying the life without being upset about Alzheimer's disease by myself. So I think we have to develop biomarker at the same time we have to develop drugs. Not just to put the money on biomarker because it's easy compared to drug development. Here, that's Alzheimer's disease now. That's a normal brain. The weight of a normal brain about between 1 kilo to 1.2 kilo. That's Alzheimer brain. Post-mortem analysis, the volume of the weight of the brain is down to 700 to 800 grams. Then that is at a PET scan analysis where you look to the glucose utilization in the normal brain. You have yellow, red, green, everything is okay compared to that. Does that mean there is a good engine, a bad engine? Okay. The key point in Alzheimer's disease is the fact that you are losing neurons. It's obvious from ear to ear. This is made mainly from neurons. And due to an oxidative stress, reduced oxygen species are involved in the killing of neurons. That's a key point. If we are able to stop this excessive oxidative stress in Alzheimer's disease, we may be able to stop the evolution of the disease. From biological studies, we know that there are a problem with amyloid plaques, which are outside of the neurons. And there is, in Alzheimer's disease, accumulation of plaques of reticulated amyloids peptide. That's outside of the neuron. Inside of the neuron, there are some tangles, neurofibrils, which are due to the hyperphosphorylation of the tau protein. What is the tau protein? Tau means tubulin associated unit. It's a protein which is related to the function of tubulin within the neuron. And we have these two, when you look to neurons, outside and inside, you have these plaques and this tangle inside of the neuron. Concerning amyloids, amyloids are generated by the fact that we are cutting an APP protein, which contains 600 amino acids. And among the 600 amino acids, there are some enzymes named secretase, which are able to produce the amyloid beta 142 and 140. On the 142, it's much more toxic than the 140. And we understand this difference of toxicity because these two peptides have a different structure. And this has been nicely analyzed recently in few papers published in Nature Structural Biology. And this is at this scale, you can see these fibrils. These fibrils of amyloids, in fact, when you try to understand the structure at the molecular level, looks like that. This is the data obtained from this NMR analysis. It looks like you have a beta sheet, 3 beta sheet, stabilized by a salt bridge between a lysine and an aniline. Okay, the lysine the aniline is here, and the lysine 28 is here. So this is a weight here. In blue, the lysine. In red, you have the aniline. And so you have these 3 beta sheets for each motif. And then you have a stacking operation due to the stabilization between these 2 amino acids. On top of that, there will be the role of metal ion. I will come on this point in a few minutes, but the key point is that. For the 142, this is the situation, and the 140 is different. This is the situation for the 140. You have a stacking, but the stacking is not as good as the stacking of 142. So it's an equilibrium. In a normal situation, we have to understand that this amyloid, we don't know exactly which are the key roles of amyloid in the brain. But in a normal situation, we have an equilibrium between this monomeric species, oligomeric species, and polymeric species. And you can go back from one form to another form in a normal situation. This is a non-freezed situation. So all these forms are in an equilibrium. In the pathogenic situation, probably there is a reduction of this equilibria, and the accumulation of the 142 is probably one of the key factors. We know that amyloid are toxic because there are many works which have been published in that field, and I'm just mention few of them. There is a connection with oligomers. Most of the people recognize that the real toxicity of amyloids is not only related to the plaque, because some people have a strong Alzheimer's disease, and when you look to the brain post-mortem, the level of plaque is not as high as it expected for where people were talking about 10 years ago. People say plaques, Alzheimer's disease. Now the key point is probably related to, concerning the toxicity, to the oligomeric form of the A-beta amyloid. There are many, and especially I would like to talk about this paper, which is a Japanese paper, which is important because when you look to the literature on amyloids and tangles, amyloids and tau protein, there were two groups of people fighting each other. The amyloid people and the tau people. And there was two different groups in two different meetings, two different conferences, unable to talk to each other. And the situation is different now. Some people understand that oligomeric form of A beta are able to contribute to the abnormal tau phosphorylation. And I think that probably these oligomeric form are triggering the tau phosphorylation. So there is a connection between the toxicity of tau phosphorylation and this oligomeric form of A beta. Nothing is divided. Things are much more complex, and they are, because these oligomeric forms are able to cross the membrane of neuron. If they are produced outside of the neuron, they are able to enter within the neuron and to target the hyperphosphorylation. Now let's go to my field, which is coordination chemistry, as you know. And I've been involved with trying to understand the role of metal ion in living system, including for modeling of cytochrome P450. But that's part of the work. I was interested by metalloenzyme and the role of redox active metal ions in living system. And when you look to this data, in red you have the titration of the accumulation of copper, iron, and zinc in the Alzheimer disease brain, post-mortem analysis. And when you look to the concentration of these metal ions, two of them, copper and iron, are redox active metal in the biological situation. You can see that compared to a normal brain, in blue, you have a real accumulation. This is a real difference. I'm not talking about 5%, 10%. We are talking about more than 5 or 6 times. This is the blood concentration. You can see that in the brain, we have much more copper than in blood. Why? Because we need a huge amount of copper for some copper enzyme like tyrosinase. Tyrosinase is on the way to go from tyrosine to DOPA. This hydroxylation of tyrosine is performed by copper enzyme. So we need copper in the brain. We need also iron, and we need also zinc. Zinc is involved in the modulation of the allosteric interaction between proteins. It's not redox active, but the coordination sphere is very flexible for zinc. It's why we have a huge amount of zinc in the brain. So if we have an excess of copper and iron, any coordination chemist interested by the role of copper and iron in living system understand that if we are in a non-controlled situation, what I mean by non-controlled situation, that means if you think about this copper and redox active copper ion, redox active iron ion in biological system, because they are redox active, they are able to produce, to react with molecular oxygen when they are in a reduced form. Copper 1 transfer very quickly an electron to molecular oxygen when copper is uncontrolled. The marvelous nature is able to control the activity of redox metal ion in living system because we are able in all living systems to control the concentration of this metal ion. That means we have system where we can transport iron to an iron protein or a metal enzyme without being perturbed by having a reduction of iron 2, iron 3 to iron 2, and so on. So, hemoglobin is able to carry molecular oxygen without reducing molecular oxygen. We have to think about that. On delivering oxygen to myoglobin without reducing molecular oxygen. In P450, we start to have a reduction of iron 3 to iron 2, and then molecular oxygen is in the cycle. But at the moment, when you start the cycle of P450, you have the substrate, which is here. And if we are doing something with a reduced form of oxygen, in fact, we have the substrate. And nature is good. Instead of reducing, is trying to have metal oxospecies and things like that. That's mean. The message is, when nature is handling redox active metal ion, is doing in the non-pathologic situation in the way that there is no non-control reduced system. When you have this accumulation of copper with amyloid, you have amyloids, copper, and you put a reducing agent, then you start to produce in vitro very quickly reactive oxygen species, and you do the titration, for example, of the formation of hydrogen peroxide. So, we can imagine that Alzheimer's disease is related, it's one hypothesis. It's related to the fact that when you have this accumulation of copper or iron in the amyloid with the short oligomers, then you start to create a catalytic system which is producing reduced oxygen species, which is killing neurons. So the hypothesis to start to design drug from the coordination chemistry point of view is to think about new chelating agent, and I will come back to that in a few minutes. Where are these copper on amyloids? You can read this very recent good review article where you have everything concerning the coordination chemistry of copper on amyloid. You have a strong binding site involving this amino acid here, aspartate, histidine, two histidines, and this is more or less the coordination of copper in this part. It's why when people are using amyloids instead of using the 142, the 140, usually you use the 127 or 28, which is at the beginning of the peptide. I was talking about reduction of hydrogen peroxide, and then on the way down to the reduction of molecular oxygen, then you have hydroxy radical, and you know that there is no way to detoxify any living system where you have an overproduction of hydroxyl radicals. So we have to stop the formation, to avoid the formation of hydroxyl radical, you have to stop the first step, the 1 electron reduction, or the 2 electron reduction where you are producing hydrogen peroxide. What about the future of metal chelators, chelating agent, as potential anti-Alzheimer drug? So we know that there is an accumulation on plaque. We can understand why the accumulation on plaque is the origin of the oxidative stress. And we are also to know that the copper is involved in the regulation of the N-methyl D-aspartate receptor via the cellular prion. That's Prusiner hypothesis, and that means copper is everywhere, more or less. Now, the key point, especially when you talk to medical doctors, it's can you design a specific chelating agent? For medical doctors, usually, most of medical doctors didn't receive any training in coordination chemistry. So when you talk about chelating agents, they say EDTA. EDTA is chelating everything. So no future for specific chelators. Because if you start to talk with a medical doctor about why some ligands are specific for copper and not, then you have to talk about d orbitals. And the medical doctors receive a short training on molecular orbitals limited to S and P. And nobody is able to talk about d orbitals. and that's a limitation. I think it's when you have a training of medical doctors. How many medical doctors are in the audience? You need to have a basic training, not only on genetics, but on every different part of chemistry, physics, and so on. Just at the beginning, with good teachers, in order to have a good teaching level and good. D orbital is a problem. Okay, let's talk about D orbital. So people are talking about EDTA, tetrathiomolybdate. How can you get tetrathiomolybdate able to cross BBB? Oh, that's because it's a polyanionic system, and we know that BBB is screening and able to will be able to stop entering of such ligand in the brain. There is another interesting story, is cleokinol. And cleokinol, and the analog of cleokinol, which is BPT2. What's the meaning of this acronym? P for prana, it's a biotech. So prana, biotech, okay, BPT, and the second, because it's a second molecule after cleokinol, which has been used as a treatment, potential treatment for Alzheimer's disease. So what is the structure of cleokinol? This is the structure of cleokinol. This molecule has been used as all intestinal antibiotics in the 50s. That means this molecule has been approved from one particular medical use. So when a molecule has been approved for one particular medical use, any medical doctors, any MD, will be able to go to the FDA agency, the US, with some experiment on animals to say, I would like to use this drug for Alzheimer's disease. So this molecule, because it was already an old molecule, has been used in preliminary experiments. Okay, for Alzheimer's disease treatment. But the bad news, it's we have to remember that cleokinol has been withdrawn from the market, especially because in Japan, people were very sensitive at the toxicity of this drug, and there were a severe reduced motricity of leg. When you say on the screen, severe reduced motricity on legs, that means there were people on wheelchairs. So carefully, this molecule has been withdrawn from the market. Then to decide to use this drug for Alzheimer's disease was very, very challenging. But development has not been very good, so the molecule has been stopped. And they designed a new molecule where they remove, in fact, this is a iodide attached to an aromatic. Any people having a minimum of basic training in chemistry understand that when you reduce such compound with one electron, you create a radical anion, and this radical anion has a possibility to cleave very quickly to the carbon iodine bond, creating a radical on this aromatic benzene, which is not very good, because this radical will able to react with DNA and so on, and will be also able to react with molecular oxygen, creating a peroxide. So for any person having a basic training in pharmacology and medicinal chemistry, nobody will design now a drug with such a structure with this allied here. So they remove. That was the design of BPT2. Unfortunately, molecule was not very active. And we work, I will show you at the end of my talk, why maybe now we can understand why this molecule was not so efficient in the modulation of copper with amyloid. These molecules are bidentate. You have a chelation of copper from this oxygen, and this one is not directly involved in the coordination sphere of copper when copper is chelated by this molecule. Okay, now we are more and more accumulation of data concerning the toxicity of cleoquinol. And in Toulouse, about nearly more than 10 years ago, we decided to make new chelating agents in order to chelate copper when copper is chelated by amyloids. And the idea was to have a tetradentate ligand in order to bind copper. Cleoquinol has only 2 binding sites. That means you need 2 ligands to have a full coordination of copper with the ligand. So we decided to have a tetradentate ligand in order to have a full coordination of copper and to extract copper from amyloid. We had patents on this molecule, which has been delivered in the United States, Canada, and Europe. Key advantage of this molecule. The first point, it's without a problem of chelation, because these molecules are aromatic, they are able to interact with amyloid and to reduce the aggregation rate of the amyloids. If you work with A-beta 142, you have to realize that it's very, very difficult to work with the monomeric form of the 42. Usually it's a mixture. You can get, by purification, the oligomer, the trimer, or the hexamers, but the monomer, you forget about that. With the 140, it's possible to work with a monomeric form. With the short amyloids, the 128, whatever it is, then it's easy to form. But 142, it has a tendency to aggregate very quickly. But if you add one of these tetradentate ligand, you reduce the kinetics of the aggregation. First step. The other thing, it's this tetradentate ligand are able to stop the production of hydrogen peroxide when you have amyloids, copper, and ascorbate, a reducing agent. Why? Because the tetradentate ligand is able to extract the copper from the amyloid. And then you have, when you have the copper with the amyloid, the reduction of the copper 2 chelated by amyloid is easy. The difference between copper 2 and copper 1 is small. That means any electron is able to reduce chelated copper 2 by amyloids in order to start to produce the reduction, the catalytic reduction of molecular oxygen. When you have this tetradentate ligand, you increase the gap between copper 3 and copper 2, and then you reduce the kinetics of the reduction of molecular oxygen. That's a good point. So we check that by looking at the inhibition of the production of hydrogen peroxide. Everything has been published in rather old papers. The other thing, it's when you have to think about full chelation of copper with chelating agent, it's much more easier to think about one ligand having 4 coordination sites than 2 ligands for 4 coordination sites. The pharmacokinetics data are different because you have to take care about one species, not 2. Okay. That's I prepare all my slides on one particular computer. I don't know if there is what problem is that. I hope it will be the only mistake. Okay. Boop. Yep. Let's talk about animal model with this slide. Most of the people, not to say 100% of the people, which are trying to develop new anti-Alzheimer drugs, are working with transgenic mice. Okay. The marvelous thing in modern pharmacology, if you have one receptor, for example, and you want to check the activity of one drug with respect to one receptor, you knock out a mice where you stop the production of this receptor and you see if there is a response or toxic effect of your drug candidate. That's marvelous. Everybody is teaching that since the last 20 years in any faculty of pharmacy of medicine. That's magic. Knock out mice for monogenic disease. Okay. Because of this system of the influence of genetics on drug design, when people are talking about development of anti-Alzheimer disease, they say, let's build a transgenic mice system where we have modification of 1 or 2 genes in order to see if we have something. Okay, so you play the game and you have modification where you play with the evolution of modification concerning the production of amyloids with mice. So you knock out 1 or 2 genes, you modify genes. You have the so-called transgenic mice model where you can modify 1, 2, maybe 3 genes, not 4 or 5, because then you are unable to have mice able to survive with more than 3 modifications of genes. And everybody is playing the game with this transgenic mice model. I play the game with this mice model. But one thing, it's want to make experiment on transgenic mice model, you have to put a huge amount of money on the table. Why? Because the cost of 1 mice, a transgenic mice, is more than 100 to, depending where you pay, 1,000 to 1,500 US dollar per mice. A normal mice in Europe, in the States, is $2-3. So that means if you want to make a simple experiment, usually it takes 1 year because you have to get these transgenic mice, and then you try few drug candidates, and at the end of the year, you pay $100,000 for just one experience, 1 year. So the system is not very productive. If you think about the way we do the evaluation of drug candidate for antibiotics, 1 week with normal mice. For anti-malaria, it's 28 days because we wait nearly 1 month to be sure that we are curing the mice. So it's very efficient, fast, efficient, and the cost is reasonable. For a startup company, such amount of money is a real problem. Even for big pharma company, because at the end, they are putting a huge amount of money on these transgenic mice without getting any new drugs. And nobody, up to now, decided to think about, are these transgenic mice model a good model or not? In my opinion, because I paid this bill, I was depressed. And they say, we need to find another system to do the evaluation of the drug. And from the data I gave to you concerning the fact that the field of Alzheimer's disease for drug design is a field where there is 100% of failure, should put an interrogation in the brain of people doing drug design for Alzheimer's disease. Okay, so we decided to shift from transgenic mice to non-transgenic mice. So what we did, if we accept the idea that short oligomers are toxic, let's try to inject in the brain, near the hippocampal zone of the brain of mice, let's try to inject oligomers of 1-beta-42. So we purify the 1-beta-42, and we inject this, the trimer and the hexamer in the brain. You can read all the detail concerning the methodology in this +1 paper, which has been published 3 years ago. So, 1 injection, and at the day 21, we check if there is any modification of the episodic memory of the mice. And the answer is yes. By 1 single injection of A-beta 142, you reduce the episodic memory of the mice by 50%. Or you can control and check the episodic memory of mice. A simple box like that, where you have a decoration of the walls. You have a grid here at the bottom. You have a camera on the top in order to look to the motion of the mice within the cage. And so what you do, it's you have the possibility from the grid to have some electricity at the bottom to get an electricity discharge on the legs of the mice. Okay, small, just not to kill the mouse, but just to say there is some electricity. So at day 21, you put normal mice in this cage. After a few minutes, so the normal mice is running in the cage in order to check if there is something to eat or something to play with that. That's the behavior of normal mice. And after a few minutes, everything is controlled and monitored. You have a beep, boop, and then electricity shock. On the normal mice receives this electric shock. So you remove the mice from the cage. You put the mice back. On the day after, you put the normal mice in the same cage. On the normal mice, remember that the day before, she received something from in the leg. So she starts to freeze. It's a freezing period. So you monitor the behavior of a normal mice which has a memory of what's happened the day before with what's happened the day after. And you have a way to monitor the loss of episodic memory. That's the episodic memory. The same memory that you are using when you put your car in the parking place. When you go after the shopping center, you say, well, I remember my car is somewhere here. Okay? We have to remember that in Alzheimer's disease, you start to check that the person has Alzheimer when this person is unable to go back after moving away 300 meters from his own street, from his own home. Not able to go back alone at home because no episodic memory. Okay. If you do that with the mice which receive the short injection, one injection of the oligomer, you have a loss of 50% of the episodic memory. This is the normal situation, the control mice. This is a mice which receives the 142 oligomers, down 50%. This is very reproductive. The experimental error is unbelievable for experiments performed on mice. How can we sure that this episodic memory loss is due to the amyloid? There is one control experiment, which is a key control experiment. It's you inject, instead of using the 142, you inject the reverse peptide, the 42 to 1. Same amount of peptide, but the sequence is different. And we know that there is no aggregation of this amyloid compared to this one. And with the reverse peptide, there is no loss of episodic memory. Okay. Then we treat the so-called infected mice with a chelating agent 3 times per week for 3 weeks, Monday, Wednesday, Friday. On the 3rd week, instead of treating the mice on Friday, you do the experiment with a cage. That's a 21-day experiment. And then you can see if your molecule is active or not. And this is one of the tetradentate ligand, which is page 1637, and you can see that we are back to the level of the control mice. With a cleokinol, more or less, but not as good. I'll say, maybe there is different, but at least here we have. And we know that the effective dose, where you get 100% recovery, is 12.5 milligram per kilo. If we go down to 6, we have 50%, so we know that the real active dose is 12 milligram per kilo. And so we are rather happy with that. And but I have to say that I had very, very strong difficulties with publishing this paper. I try Science, Nature, PNAS, and it has been accepted in PLOS with very, very difficulties. Why? Because most of the people say, okay, we don't have any problem with your data. We trust your data. But, and the but was killing the paper. You need to prove that your system is active on the transgenic mice. Okay, then you say, how can you make a transgenic mice model of the copper interaction? How many genes are involved with copper carrier proteins and things like that? 1, 2, 3, 4, 5, 6, probably 15 genes. How can we get a transgenic mice model for this copper story? And if you treat the transgenic mice model with this tetradentate ligand, you get more or less the same level of activities and memantine on the transgenic mice model. So, but I think that the feeling that with memantine on the transgenic mice model, any drug is active more or less. So, so that was. And I get the authorization to publish the paper by the editor-in-chief because one of the referees was so bad, with so stupid argument, that the referee, the editor-in-chief, accepted the paper. Why? The bad argument was about the toxicity. I have one paragraph of the toxicity. I will show you the data concerning this molecule. And I was making the difference between the two toxicity, between cleoquinol. And the referee said, well, your molecule is less toxic, but you know that it's not a problem in drug development. So I say, well, I send the paragraph of the FDA administration concerning toxicity of drug. I say, what is drug development? It's a balance between activity and toxicity. And what we need for a good drug, it's a good difference between activity and toxicity. So we want to improve the activity, reduce the toxicity. So I copy 10 lines of the FDA about that. It's obvious for anybody interested by drug development. I say, well, this referee is not talking about a good point. He's stupid because he's running against FDA. And nobody is allowed to run against FDA, guidelines. FDA guidelines is, magic. So nobody touch FDA guideline. And the editor-in-chief say, okay, I accept your paper. So it's, in my opinion, the first good paper where we publish a non-transgenic mice model to check the loss of episodic memory. Since that, people ask me, how can you prove that the episodic memory loss is related to amyloids and blah, 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 and I say, I don't know, but these are facts. Now, people which are good about neurological studies in the brain will do the job for the future. But at least we have one model, which has been reproduced now in other labs, which is a good point. If you are the only person developing one model, that's not. But now there are other people. Good. So what about the toxicity of this tetradentate ligand? By oral administration, we don't kill mice at 450 milligrams per kilo. We have to remember that the active dose is 12 milligrams per kilo by oral administration. I forgot to say that. So that means we have a real window safety. And even if you have IP injection, we have still a good difference between our molecule and the cleoquinol. And so that's a summary. We have a molecule which is relatively easy to make. EMS negative. We don't expect to have mutations. The cardio safety is okay. There is no toxicity on mice. There is no acute toxicity and no oculotoxicity. Cleoquinol is toxic, and you can check the toxicity on the eyes of the mice. There are problems. They are getting red eyes after a few milligrams, so toxic. Now, the key point to understand the activity of the molecule compared to cleokinol and BPT2, it's to understand, is this molecule able to extract copper from amyloid? The answer is yes, because we inhibit the production of hydrogen peroxide, and we check that by just making some regular experiment in coordination chemistry. So the key point is to have a demonstration of this is copper going to amyloid. That's a complex between copper and amyloid. Then we have a so-called mediating ligand, this tetradentate ligand, able to extract copper to produce this tetradentate copper ligand. And the key point, if we want to have a good drug, is to release the copper in order to give the copper back to a copper carrier protein. Because we don't want to deplete brain from copper. In my opinion, it's a misregulation of copper circulation, which is the cause creating Alzheimer's disease. So we need to put again copper in the normal circulation of copper or copper trafficking in the brain, because we need copper for tyrosinase, for example. So we need to have a drug entering in the brain, extracting the copper from the amyloid, and putting back the copper in the circulation inside, outside of the neuron, normal circulation. So we have to demonstrate in vitro at least all these different steps. I'm not going to enter in all the details, spectroscopic detail, but the conclusion is we have effectively a ligand which is able to do that. We published one paper last year, and we published a second paper. This is the structure of the ligand with copper, and we have now been able to demonstrate that our ligand is able to do the job, and we understand also why cleokinol is not doing the job, or why BPT2 is not doing the job. Because when you look for cleokinol with copper and amyloid, what is doing cleokinol? Two coordination sites which are available on the copper linked to amyloid. You have cleokinol making a ternary complex. Amyloid, copper, cleokinol. Cleokinol is missing the two other coordination sites in order to extract and to make a stable complex. That's probably why there was a failure of these molecules as chelating agent when they have been used. So this is a summary of what we want to do. We want to stop this pathological situation. That means we want to extract copper from amyloid with a good ligand. And then on the reduction, we have the reduction of the copper to ligand, which is able then to release copper 1 to a copper protein at the apo form. When this copper carrier is not involved with copper. And then we are going to put back copper in the trafficking of normal situation in green. So we want to move from the red situation to the green situation. Everybody should be green. Okay, that's a green situation. But we published that in particularly this paper, which is in press. We have now the full demonstration of that. We summarize that in a recent account of chemical research. And now I have to thank all the people which has been involved. Oops. Sorry. Can I get the acknowledgement picture? Slide. I want to get the last slide. Go like that. Because now I will go bum, 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 bum. Down, 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 down. This is a key slide because it's the name of the people which has been involved. Anne Robert was involved in this topic since 2011 with Michel Guyenne. And I created the non-transgenic mice model with a colleague in biological lab, which was near the retirement period. So we decided both to do this non-official model for Alzheimer's disease. So we took the risk, and it was a nice period when you have the chance to talk with a colleague and to make something new which has not been made before, and where you are taking risk. Oh, no, we never received any support from any official agency for that because we are out of the scope, and so that's a key point. So it's a lesson for funding agency. You have to trust people from time to time. If you put the money on people who are just able to reproduce, which has been done in other labs, okay, you make papers. But if you want to improve innovation in a country, you have to trust people which are funny ideas from time to time. That's another lesson. So many thanks to Jean-Michel Lassalle and Jonathan Secom, which was a young PhD who did the experiment. Then all these people from Polymed working on the project. And this project now is going on in mainland China in the University of Technology of Guangdong in Guangzhou, where I have a position as a professor for 5 years. There was some financial support from Polymed, CNRS, Région Midi-Pyrénées, and the Guangdong province and GDTU, and I would like to thank for your very kind attention. It was a real pleasure to talk to you today.
Would you mind to answer questions?
Oh, absolutely.
Thanks so much for the exciting talk. A quick question. I totally understand how the cleoquinone can extract the copper-2 from the A-beta, but once it's released, how can you avoid
that? That's the point. I tried to. The cleoquinone, in fact, is unable to extract copper. Cleoquinone is making a tertiary complex. So you have copper with the amyloid, and the cleoquinone is making a tertiary complex. So you have the 4 coordination sites of copper occupied 2 by the amyloid, 2 by the cleoquinone. And I was too quick at the end, but we published that. We have been able to have the mass spectrometry data of the amyloid, copper, and cleoquinone. And that's the difference because when we extract with the tetradentate ligand, we really extract copper from the amyloid. So in my opinion, cleoquinone is not a good ligand for modulation of copper from amyloid. So you need a tetradentate ligand.
Thanks very much for your talk. I have 2 questions. The first question is, since we know there's also zinc and iron involving A-beta binding, so once your ligand extract copper, do you think the A-beta will further interact with iron and zinc and then cause problem?
One point that I forgot to tell during my talk, it's this ligand is specific for copper. So we don't touch iron, we don't touch zinc. That's a marvelous of coordination chemistry. Now I can't answer your question at the moment. We don't have. My view is if we remove copper from 142 amyloid, then we may be able to have to go back to an equilibrium situation between the oligomer, the polymer, and the monomer. And then these amyloids, when after removing the excess of copper, may be able to go back to a normal situation. Maybe copper will be replaced by iron or zinc, I don't know. But In order to answer your question, we will need in the future for a specific iron chelator in order to distinguish between the role of copper, the role of iron. For zinc, forget about a specific zinc copper because we know that we need a huge amount of zinc for the interaction between normal protein and the non-pathologic situation. But it will be nice, in fact, to have a very specific iron chelator, and at the moment, I tried to do that, but I've not been able, and I don't know if somebody has been able in the world to have a iron-specific copper chelator compared to other chelators.
Okay. Maybe I'll leave the second question to others. Okay.
Thank you. Very nice talk. So I'm interesting to know that whether it's a copper level increase happened earlier or it's oligomer formation happened earlier. You have shown the data that Alzheimer patient got a higher copper content, but how about early onset Alzheimer patient?
It's
Do you have the
difficult
data
to answer your question because all the information concerning the accumulation of copper and iron in brain are related to post-mortem analysis. And so you get the answer after 8 years of the disease, more or less. And we don't have a good way to monitor the accumulation of copper during the evolution of the disease. So if people are trying to have a relation between the level of free copper in the blood, Rosanna Squitty is trying to do that in Roma, but it's difficult.
Yeah, but if it is possible to have data from early onset AD patient, then probably that can prove that oligomer happened earlier.
But for the early stage of the disease, how can you get information on copper coordination accumulation in plaque from the brain?
But also after post-mortem assay, but those patients are early onset. I mean, they have genetic linkage with A beta overproduction.
Well, but it's the key point is to get extraction of the plaque with copper in order to monitor the concentration. You can do that on normal brain on a person. You know, you can do that on animals, but not on people.
After all these patients die, they examine their brain.
That's it. Yeah, but you get data at post-mortem situation. It will be nice. It will be nice to have a way to monitor the copper accumulation in amyloids during the evolution of the disease. If we will be able to have a titration or way to collaborate, for example, free copper in blood with a label that, it's a brain disease. So we don't get biopsy easily from brain. It's not like a regular, it's a brain disease. It's a problem. There is a barrier.
Hello, yeah. Yes, very nice talk. So in principle, in your mouse model, you could have tested out your agent is a preventive agent. So what did you find?
I think in fact, our model, a non-transgenic model, is a model of the early stage of the disease. Okay, so if the failure of the transgenic mice model, it's a model of the last stage of the disease. In all this transgenic mice model, you have a reduction of the neuron, you have a thing. So there is no way to go back. After you kill the neuron in the brain, people are talking about regeneration of neuron in brain, but nobody's sure about that. So, so our model is a model of the early stage. So I think it's why I like this model.
Well, the last question.
Merci. And after the chelating agent extract the copper from the amyloid beta, is it remaining aggregated, or does it refold? In another way, is the source of the toxicity come from occupying the copper or the aggregation its own?
Well, we never did experiment to answer your question.
For example, if
But it will be possible to set up experiments.
does
on
the
that.
Congo red fluorescence or
We,
thioflavin T
Yeah,
of
yeah.
the brain tissue change?
It has to be done. We never did that. We just check the inhibition of the production of hydrogen peroxide, but we never check if we reduce the aggregation of the A beta count 42. It has to be done.
Well, apparently the talk raises lots of interest and questions to be answered. Anyway,
Everybody is interested,
yes,
you know.
yes. So please join me to thank President Meunier for his wonderful talk.